Welcome. We're going to be going over the Heredity chapter. My name is Susan, and heredity is simply the passing on of physical or mental characteristics from the parent to the offspring. So, who do you have to thank for your hair color, your eye color? How about your intelligence level or your ability to learn? What about your propensity to certain types of cancer, diabetes? Before we delve into heredity, let's give a moment of thanks to George Mendel. George Mendel published his work on inheritance in 1865. So when the Civil War was wrapping up, he was working with pea plants. He was figuring out how characteristics are inherited. I guess that's better than getting shot. Now, once his work was published, it was actually ignored for about 30 years, but then adopted as the foundation for modern genetics that we know today. We'll start with chromosomes. We all have them. Humans have 23 pairs of chromosomes, so that's a total of 46 in our body. It's just the form that DNA takes, so now it looks like something. As we continue to go on with this chapter, we'll be reviewing some terminology. We'll start with genes. If you take one chromosome, you take a section out of it, that's a gene. It's a specific section of DNA on one chromosome. Genes determine things including the ability if you can smell asparagus pea or not. But we'll work with something more basic, eye color. So the gene says, let there be eye color. Let there be eye color. There are alleles that will dictate what type of eye color. So the gene demands eye color. There's an allele for blue eyes. There's a brown allele for brown eyes. We've got a lot of variety of eye colors. We've got gray, amber, green, hazel, even red. And I'm not talking Edward Cullen red. In this case, the red is actually due to albinism. So you may have inherited one allele from your mom that says that you will have blue eyes. You also may have inherited another allele from your dad that says you'll have brown eyes. That's actually a common occurrence. And if it was that easy, we'd all be walking around with two different colored eyes, like David Bowie. What actually says which allele will win the battle of alleles is which one is dominant, which one is recessive. Some more terminology that we'll come back to even later, but let's talk about what that means. Dominance just means that it's the allele that's going to present and show itself. And it's going to hush the quiet recessive allele. So the dominant is more like a stage hog wanting all the attention and glory. And the recessive is like the wallflower. Just because an allele is recessive doesn't mean it is uncommon or rare. Actually, quite often it could be the opposite. You can have dominant genes showing up all over the place, and they can actually be rare. Again, just stage hogs and wallflowers does not mean that necessarily there's one or more less of each type. So we started talking about chromosomes, touching on that different sections of a chromosome is your gene. Homologous chromosomes, what that means is that out of humans' 23 pairs of chromosomes, 22 pairs, number 1 through 22, the first 22 are homologous, and they are called autosomes. They're similar in length, and they have the same gene sequence. Now, while similar, they're not identical, but that means homologous. So for our 23 pairs of chromosomes, that is our human karyotype. A karyotype just means the number and appearance of chromosomes in the nucleus of an eukaryotic cell. The term is also used for the complete set of chromosomes in a species or an individual organism. We're humans, so we have 23 pairs of chromosomes, and our karyotype is 23 pairs or 46 total chromosomes. The 23rd pair, the final pair, is the sex chromosomes. They're the sex-determining genes. Males will be XY and females will be XX. Chromosomes, genes, I'll throw another word at you. Genome, the genetic material of an organism. The complete set of DNA including all of its genes. So one genome contains all the information needed to build and maintain that organism. So it's even bigger picture. Now, we mentioned alleles before. If you have a gene dictating, let there be color, the allele will say, which type of color? This is actually pretty awesome because this causes variation. And all the variation makes a population more genetically diverse. Otherwise, we'd all look alike, think alike, be exactly alike, and that's boring. Let's tackle another concept, heterozygous versus homozygous. So, going back to the example of your parents giving you eye color. Let's take your mom, for example. Just like you, your parents are humans too, and they both inherited alleles from their parents. So let's say your mom, she could have randomly gotten a recessive gene from her mom, maybe your Mimor, and a dominant gene for a blue eye color from her dad, your Pawpaw. That would be considered heterozygous. Hetero meaning one of each, both. So you've got one recessive, one dominant, heterozygous. And your father may have inherited two dominant alleles, one from his mother, one from his father, for giving blue eyes. That would be two of the same, homozygous. Homo equals same. So that's what those terms refer to. Working with that recessive and dominant genes, mother's side being heterozygous, father's being homozygous, we're going to fill in a Punnett square. This is a simple, basic Punnett square, and it just determines that when these two mate, mother and father, what chances the offspring have of having what kind of eye color. So you bring down into each box for this row, bring down your dominant allele indicating blue eye color. We're going to bring it down here as well, BB. Now you're going to fill in, for this row, your recessive. Remember, we're talking about just bring this straight over, bring over your dominant B here from the mom. So it looks like the way this one worked out, it's not always going to be that case, but this one worked out that about 50% of the kids can have dominant for blue or recessive for brown, dominant for blue, recessive for brown, and two dominants. So they'll actually all end up having blue eyes. Could have been different if maybe we had another brown recessive gene in here somewhere. Let's say, for example, this was a recessive allele for brown eyes. It would have changed the ball game for making blue eyes, brown, blue, and blue still. So instead of 4 kids having blue eyes, we just changed the game. 3 would have blue, 1 could have brown. That's how your Punnett square works. So now that we talked about dominant and recessive, they help factor in your genotype versus your phenotype and what that is. Genotype is an organism's genes, the set of genes that it carries. The phenotype is how those genes are expressed, what we see. So for example, you see blue eyes or brown eyes. That's the phenotype, how the genes present themselves. On me, you see my skin color, you see my hair color. Those are my phenotypes expressing the genes that I inherited. Now, genes can variate, and new alleles can be formed by some things such as chromosomal crossing over and recombination during the sexual production phases. This topic is called genetic variation. This is a pretty cool one that we're going into because of what can come of it. The sexual sources of genetic variation, there's 3. We'll start with segregation and independent assortment. Back to Mendel. Remember, he was publishing his work just as the Civil War is wrapping up? And his experiments led to something called the law of independent assortment. It describes how different genes independently separate from one another when reproductive cells develop. What that means is that traits are transmitted to offspring independently of one another. One allele that a gamete receives for one gene does not influence the allele received for another gene, so no peer pressure. They're all just doing their own thing. Now, according to Mendel's other law, the law of segregation, that means that only one of the two gene copies present in an organism is given to each gamete that is made. Remember, our gametes are the sex cells, the sperm or the egg. And what is given to which cell is just random. A second source of genetic variation is crossover of homologues and gene recombination. This is actually more fascinating than it sounds. We're going to start with linkage. And our example for linkage is if you inherit an allele for blue eyes and an allele for oval-shaped eyes. Well, instead of getting each allele separately, you're going to get these alleles as a combo unit. That is what linkage is, that they're linking up. Okay, so the next concept of crossover, I drew up on the board to maybe hopefully make it easier. So step one, you have two homologous chromosomes. They're aligned. The second step in crossover is you see how they come together and they literally cross over. The third step is that when that happens, they'll each take a piece from each other, what crossed over. So you see how this chromosome has taken from the black and the black took from the blue. Those two are called recombinant chromosomes. The two that weren't touched are non-recombinant. Crossing over actually creates new combinations of genes in the gametes that are not found in either parent, contributing to genetic diversity. And that's a really cool concept. So you'll end up with stuff that you may never have inherited. Crossing over also frequently occurs during the prophase 1 of meiosis. Prophase 1. The third and final thing is recombinant chromosomes. Just like the drawing I mentioned, the two chromatids that did not participate in crossing over, well, they'll still have the parental combination of alleles, and the other two will be recombinant. They're recombined. It's in the name, so don't go insane. Oh, side fun fact. So most recombination is going on naturally, occurring in the body during the meiosis of sex cells, but some recombination is done in the lab and will produce things such as vaccines and antibiotics. Now, the third and final type source for genetic variation is actually random fertilization. This refers to the fact that when two people mate, each is capable of producing over 8 million potential gametes. The random chance of any one egg and sperm coming together is a product of these two probabilities. So there's over 70 trillion different kinds of combinations of chromosomes in a potential offspring. What that means is that to exhaust every possible type of kid you'd have to have over 70 million kids with just one other person. Don't even bring in other people. Then our numbers are going to be crazy. So forget what the Dugars with 19 and counting have going on. You not only have your own reality TV show, they probably devote a whole network to you. Okay, so let's talk about types of inheritance. You've got something called lethal dominant genes. Lethal genes can be either dominant or recessive. They can cause the death of the organism that carries them. So they can be rare because the allele often lasts only one generation and is not transmitted. They're usually the result of mutations in genes. There's also disorders that can be inherited. One type of disorder is achondroplasia. Chondroplasia is short-limb dwarfism. The word actually means without cartilage formation. Cartilage makes up much of our skeleton during early development, so if you have achondroplasia, then your cartilage didn't convert to bone. It's a gene mutation. Another disorder that can be inherited is Huntington's disease. The allele for Huntington's disease is dominant, while the allele to not develop it is recessive. Other things that can influence gene expression, the environment. Environmental influence of gene expression. Things such as temperature, light, chemicals, infectious agents, even your age, your gender. You've always heard that women in their 40s have a higher probability of giving birth to a baby that has Down syndrome because age affects that 21st chromosome. So they have a higher probability than, say, a woman in the 20s. Drugs, chemicals, another good reason to not do drugs. Don't jack up your baby. Even pot, more research is needed. But the National Institute on Drug Abuse says that research has shown that marijuana use can lead to problems with neurological development of a fetus. That's a kind of a big, scary thought. In school, children that were exposed to marijuana in the womb have been more likely to show gaps in problem-solving skills, memory, and the ability to remain attentive. So, why risk making your kids stupid? Toke up before and after pregnancy? Not during. Another type of inheritance with genes is non-traditional. So inheritance patterns that do not fall into the chromosomal, single gene, or the multifactorial categories qualifies as non-traditional inheritance. And Fragile X syndrome is one of these examples. Fragile X is an inherited condition characterized by an X chromosome. Children that are affected by this condition usually tend to have limited intellectual functions. The X chromosome is abnormally susceptible to damage, unfortunately. With all these things that could be inherited that are problematic, thankfully in the modern day and age, there's screenings for this kind of stuff. So some examples of that is genetic screening. They've got something called carrier recognition, and that's pretty cool because it's a type of genetic test that can be used to determine if a person who is a carrier for a specific autosomal recessive disease is going to pass it on or not. The testing is often done, obviously, by people who are considering becoming parents, and they want to determine the risk of their child inheriting a genetic disorder that they carry. So they kind of know something's in their family, and they can find out the probability that their child is going to have it. This actually leads into pedigree. If a male and female are both unaffected by genetic disorder, but they're both carriers for it, well then, each of their children, there is a 25% probability or 1 in 4 chance that the child will get the inherited disease trait. That is called a typical pedigree in which a single individual is affected by genetic disease. So what's your pedigree? Do you know it? Another kind of testing to weed out things and find out more about the fetus? Fetal testing, amniocentesis, and chorionic villi sampling. Amniocentesis is done with the amniotic fluid, and it can tell chromosomal abnormalities, fetal infections. It tests for neural tube defects. That's really important because a common neural tube defect is spina bifida. It can detect for Down syndrome, cystic fibrosis. Now the other one, chorionic villi sampling, that's a test in which the small sample of cells is taken from the placenta, where the placenta attaches to the wall of the uterus. So that's the difference between the two, taking from the amniotic fluid or taking from the placenta. Let's end this chapter on a good note, looking towards the future. Human gene therapy. I like this topic. So human gene therapy is an experimental technique that uses genes to treat or prevent disease. In the future, human gene therapy may allow doctors to treat or prevent a disease by inserting a gene into the patient's cells. This could be in place of drugs, in place of surgery, instead of radiation, instead of chemotherapy. So we're just on the brink of this. Basically, it's a whole new world out there. And while our grandparents saw the advent of the color TV in their home, you guys get human gene therapy. Not exactly fair. Genetics is everything. It's in every living thing around us. For us, how we look, how we think, how we behave, it's all affected by genetics. So if your genetics are jacked up, you're screwed. You can get really unlucky, or you can be Chris Hemsworth.